Hello, my name is Asha Goodman, and I'm a PhD candidate in the Dinsdale Lab and the Forsberg Lab at San Diego State University. And I get to research sharks. Sharks are so cool for a number of reasons. One is that they have no bones in their body. The other cool thing is that they were here before trees. And so the sharks that I research migrate up and down the coast of California. And when they aggregate, specifically off the coast of San Francisco and San Diego, I have the opportunity to sample them. But they're also residing at the Birch Aquarium. And so what am I sampling? I'm actually interested in two different things. One is their blood, more specifically the molecules in their blood. And this includes vitamins, sugars, and hormones. And all the metabolites in this system constitute the metabolome. And so these are really important. Let's just picture these as cells in the host for cell signaling, energy conversion, and maybe understanding if there's a disease or infection occurring in the host. The other thing I'm interested in is sampling the skin of these sharks, specifically looking at the microbes on the skin. This includes archaea, bacteria, and even viruses. And so all these microbes constitute the microbiome. And using shotgun metagenomics, I can sequence their genomes. So I use different colors to illustrate how when we take the genomes of these bacteria and cut them up, we get fragments. And this will become important later. And with this information, we can see who's there and what they're doing. The cool part is I have two populations I'm interested in, those in the wild and those in captivity. And right now my project is focused on how captivity regulates both the microbiome and the metabolome of these sharks. And so what we found is that the metabolome isn't significantly impacted, so this includes stress hormones or other responses of the shark. The other thing we found is I look at the biodiversity of the microbiome. And if we were just to imagine these are different species of bacteria, do these shift under captive environments or wild environments? And we found that in both the captive and the wild environments, they're not negatively impacted, which is a great win for conservation, especially because these sharks are really popular sport fishing targets. The last thing I do in my project, as of right now, is assembling all of these fragments into one genome. So let's just imagine I take all the fragments of this color and line them up to become one genome, one giant contig. So this constitutes a mag. And the great part about mags is now we have an entire species. And so we can either confirm who's there and what role they play, and find novel associations with hosts, or we can find entirely unknown bacteria that haven't been annotated or sequenced yet. And we've actually found a novel association with leopard sharks that persists across captivity status and has been the first of its kind to be associated with vertebrate hosts. So thank you for hanging out with me. That's where my project is right now.